Chapter 9
by Catalina, Matt and Lynn.
9.1.1 Draw and label plant diagrams to show the distribution of tissues in the stem and leaf of a dicotyledonous plant.
Check out those cool diagrams.
9.1.2 Outline three differences between the structures of dicotyledonous and monocotyledonous plants.
Monocots, parallel venation in leaves, reproductive, flower, parts in multiples of 3 seeds with 1 cotyledon, vascular bundles scattered throughout stem, trend to have fibrous root system, pollen grain with 1 opening dicots, net-like venation in leaves, reproductive, flower, parts in multiples of 4 or 5, seeds with 2 cotyledons, vascular bundles and ring-like pattern in stem, trend to have taproot pollen grain with 3 openings.
9.1.3 Explain the relationship between the distribution of tissues in the leaves and the function of these tissues.
Xylem bring water to replace losses due to transpiration, and inorganic minerals from the soil. Phloem transports products of photosynthesis out of a leaf. Stoma, the pore that allows CO2 for photosynthesis to diffuse in and O2 to diffuse out. Guard cells, this pair of cells can open or close the stoma and so control the amount of transpiration. Upper epidermis, a continuous layer of cells covered by a thick waxy cuticle. It prevents water loss from the upper surface even when heated by sunlight. Lower epidermis is in a cooler position and has a thinner waxy cuticle. Spongy mesophyll consists of loosely packed rounded cells with few chloroplasts. This tissue provides the main gas exchange surface, so must be near the stomata in the lower epidermis. Palisade mesophyll consists of densely packed cylindrical cells with many chloroplasts. This is the main photosynthetic tissue and is positioned near the upper surface where the light intensity is highest.
9.1.4 Identify modifications of roots, stems and leaves for different functions, bulbs, stem tubers, storage roots, and tendrils.
Bulb equals stem modification consisting of enlarged bases of leaves, aids in food storage. For example, onion, stem tuber equals horizontally growing stem, aids in carbohydrate and starch storage. For example, potato, starch root equals specialized root for storing large volumes of carbohydrates and water. For example, carrots. Tendrils equals leaf modification to aid and support in climbing, able to coil around objects during growth through a thigmotropic response. For example, morning glory tendril.
9.1.5 State that dicotyledonous plants have apical and lateral meristems.
Dicotyledonous plants have apical and lateral meristems.
9.1.6 Compare growth due to apical and lateral meristems in dicotyledonous plants.
Apical meristeme exists at the root and shoot tip, and is involved in plant elongation. Lateral meristeme exists in cambium tissue. Growth in lateral meristeme increases the thickness of the plant.
9.1.7 Explain the role of auxin in phytotropism as an example of the control of plant growth.
Auxin is a plant hormone which elongates cells when a plant is exposed to a light source. The auxin migrates away from the source. In this way, the side of the plant farther from the light elongates, bending the plant toward the light source.
9.2.1 Outline how the root system provides a large surface area for mineral ion and water uptake by means of branching and root hairs.
Root hairs provide a large surface area for mineral ion and water uptake. Cortex cells absorb ions that are dissolved in water, drawn in by capillary action through cortex cell walls. Branching increases quantity and area roots can absorb ions from.
9.2.2 List ways in which mineral ions in the soil move to the root.
Plants absorb potassium, phosphate, nitrates, and other mineral ions from the soil. Active transport pump ions into the roots. Root hairs provide large surface area for ion uptake.
9.2.3 Explain the process of mineral ion absorption from the soil into roots by active transport.
Integral proteins transport minerals from the soil into roots through active transport. Once the minerals have crossed over into the plant, they attract water through a concentration gradient.
9.2.4 state that terrestrial plants support themselves by means of thickened cellulose, cell tuga, and xylem.
Terrestrial plants support themselves by means of thickened cellulose, cell tuga, and xylem.
9.2.5 define transpiration.
Transpiration, loss of water vapor from the leaves and stems of plants.
9.2.6 explain how water is carried by transpiration stream, including the structure of xylem vessels, transpiration pool, cohesion, and evaporation.
Transpiration causes a flow of water from roots to stem and leaves. This movement is called transpiration stream. 1. Evaporation of water from spongy mesophyll cell walls of leaves. 2. Evaporated water is replaced from xylem, pulled out of the xylem through mesophyll pores by capillary action. 3. Low pressure is created inside xylem vessels when water is pulled out. Transpiration pool. Xylem vessels contain long, unbroken columns of water where the pressure is transmitted across. 4. To equalize the pressure, water travels up the vessels through its property of cohesion.
9.2.7 state that guard cells open and close stomata to regulate transpiration.
Guard cells open and close stomata to regulate transpiration.
9.2.8 state that the plant hormone abscisic acid causes the closing of stomata.
The plant hormone abscisic acid causes the closing of stomata.
9.2.9 explain how abiotic factors affect the rate of transpiration in a typical terrestrial mesophilic plant.
Light. The intensity of light increases or decreases the rate of evaporation of water from the top of leaves, causing an increase or decrease in the rate of transpiration. Temperature. High temperatures increase the rate of evaporation of water from the top of leaves, resulting in an increase in the rate of transpiration. Conversely, low temperatures decrease the rate of evaporation of water from the top of leaves, resulting in a decrease in the rate of transpiration. Wind. High wind also increases evaporation by allowing more air molecules to collide with the water molecules on leaves, resulting in an increase in evaporation of water and transpiration. Low wind results in more stagnant, saturated air around the stomata, which decreases evaporation and transpiration. Humidity. High humidity decreases the rate of evaporation and transpiration of a plant.
The 9th of February 2010 outlined 4 adaptations of xerophytes that help to reduce transpiration.
Spines instead of leaves, reduce rate of transpiration. Thick stems contain high amounts of water storage to sway. Thick waxy cuticle. Vertical stems absorbs sunlight late and early in the day but not midday when sun is too intense. Wide branching and shallow root systems absorb water closer to the surface that would be otherwise evaporated before reaching roots.
The 9th of February 2011 outlined the role of phloem in active translocation of biochemicals.
Phloem have sieve tubes that transport organic compounds. Golem cells develop into sieve tubes by breaking down nuclei and cytoplasm, making large pores in their end walls to allow a flow of sap. The plasma membrane pumps organic compounds into sieve tubes using ATP. This creates a high concentration of solute, causing water to diffuse and resulting in a positive pressure gradient, allowing organic compounds to be pumped anywhere in the plant.
9.3.1 Draw and label a diagram showing the structure of a dicotyledonous animal pollinated flower.
Diagrams are so fantastic.
9.3.2 Distinguish between pollination, fertilization, and seed dispersal.
Pollination, transfer of pollen from anther to stigma. Fertilization, fusion of male and female gametes in the ovum. Seed dispersal, the spreading out of seeds, fertilized ovules, contained within fruits, which develop from ovaries, by various means.
9.3.3 Draw and label a diagram showing the external and internal structure of a named dicotyledonous seed.
Yeah. Memorize that diagram.
9.3.4 Explain the conditions needed for the germination of a typical seed.
Abundance of water rehydrates dry tissue. Oxygen. Aerobic cellular respiration before photosynthesis can occur. Suitable temperatures. Germination involves enzymatic activity and digestion of starch and cellulose synthesis. If temperatures * fall outside of temperature ranges for these enzymes, germination does not occur. This causes seasonal germination in many places. Seeds vary in their light requirements and, therefore, this factor need not be included.
9.3.5 Outline the metabolic processes during germination of a starchy seed.
After absorbing water, the growth hormone, gibberellins, is produced in the embryo scotolitins. Next, amylase, a digestion catalyzing enzyme, is produced to break starch down into maltose, which is diffused to the embryo for either aerobic cell respiration as an energy source, or for growth.
9.3.6 Explain how flowering is controlled in a long day and short day plants, inducing the role of phytochrome.
Phytochrome is a pigment, which exists in two interconvertible forms. Puerto Rico, the inactive form of phytochrome, absorbs red light with a wavelength of 660 nm. After absorbing the light, it turns into PFR. PFR, the active form of phytochrome, absorbs far red light with a wavelength of 730 nm, and is transformed back to Puerto Rico rapidly after the absorption of light. In normal daylight, there is much more red light than far red light, so phytochrome exists more in the form PFR. Thus, in darkness it reverts back to Puerto Rico. Enough PFR remains in long day plants at the end of short nights to stimulate flowering.
The end.